Is most of our DNA junk? Welcome to Critical Thinking Scan, where we look at how you can think about any faith-challenging message and arrive at a biblical, logical conclusion yourself. I'm Patricia Engler, and today let's look at the claim that our genomes are largely junk. Less than 2% of our DNA seems to code for proteins, so many researchers believe that the other 98% is a collection of useless evolutionary leftovers. I heard that message in one of my classes. But is it true? Let's think about it using the 7 checks of critical thinking. Number 1, check scripture. Genesis says God created everything very good, so biblically we wouldn't expect our DNA to be mostly useless and certainly not left over from evolutionary ancestors. However, Genesis also reveals that creation didn't stay very good because of sin, so we wouldn't be surprised to see some DNA lose its function or to see some extra DNA accumulate, for instance through copying mistakes. Jumping down to check number 5, check for propaganda, we can apply the question, is this message true or false, because to ask, is it true that non-coding DNA is junk because we don't understand its function? No. Making claims about DNA's function or lack of function based only on what we don't know would be an appeal to ignorance fallacy, which we examined in episode 33. On to check 6, check the interpretations, we can separate fact from assumption by asking, what's the observational science surrounding junk DNA? The fact we're observing in the present is that less than 2% of the genome encodes proteins. The interpretation of this fact based on evolutionary assumptions is that the other 98% must be junk from our ancestors. Well, what's an alternative explanation from a biblical perspective? Biblically, we'd expect that DNA created very good probably has important functions, even if we're not yet fully aware of them. So what's the other side of the junk DNA story? What other facts make more sense when explained through a biblical lens? For one thing, researchers with the ENCODE Project, which stands for Encyclopedia of DNA Elements, reported in 2012 that the vast majority of the human genome is transcribed into RNA, which plays a role in which genes get expressed in which cells under which circumstances. That's what lets cells with the same genes turn into different tissues during development, like liver or lung, depending which genes are switched on or off. A commentary in the journal Nature concluded, 80% of the genome contains elements linked to biochemical functions, dispatching the widely held view that the human genome is mostly junk DNA. This statement caused enormous backlash because, as Scientific American observed, it's evolutionarily impossible for so much of the genome to truly function for human health. Critics also argued that just because 80% of our genome is linked to biochemical activity, that doesn't prove the RNA is functionally necessary, although we wouldn't expect cells to waste much energy transcribing messages which aren't important. So instead of defining junk DNA as DNA which isn't transcribed, some evolutionists define it as DNA which isn't affected by natural selection. They also define functional DNA only as DNA which seems to be conserved by natural selection, meaning that just 3 to 15% of the genome would be considered functional. However, other researchers have pointed out the problems with only using evolutionary definitions of function. For instance, one team said that disease-relevant regions need not be selectively constrained to be functional. Besides, a 2018 study found that actually 80 to 95% of the human genome is influenced by some sort of selection. The authors carefully stated that they weren't implying all that DNA is functional, because they thought their findings reflected indirect impacts of selection acting on other parts of the genome. But their findings also make sense if the whole genome really is functional. And as further ENCODE data released in 2020 documents, researchers are still uncovering more and more of that function all the time. In a recent Nature article, ENCODE scientists wrote, "It has become apparent that by virtually any metric, elements that govern transcription, chromatin organization, splicing, and other key aspects of genome control and function are densely encoded in many parts of the human genome sequence." So, observational science continues affirming that the genome is not mostly evolutionary junk, but rather incredibly well designed. We'll look more at objections to ENCODE's conclusions next time. Meanwhile, for more on how to think critically about faith-challenging messages, you can access my other CT scan videos packed with tactics, tips, and tools that helped me as a Christian student at Secular University. Thank you for watching. Hey, it's Patricia. Just wanting to let you know that if you like these videos, are on board to share the message of biblical authority, and want to give, you're welcome to help Answers in Genesis produce more content like this and equip more people to defend their faith by making a one-time donation or becoming a monthly supporter by clicking the link below. Thanks so much.